Yeah. Good morning, guys. Arshia, Samvarna, Abhiniti, Nivedita. Good morning. So, is the board is little clear? I mean, is it clear? I'll be not writing anything, letters and all. Just I wanted to show some structures. That's it. So, if it is clear or not, you please let me know. Is this clear? Are you able to see? Shamili, good morning. Okay. Just a minute. Yeah, okay, fine. So today we're going to discuss about two important chapters. One is cell structure and function. So different cell organelles we'll be talking about, right? And we'll be talking about the cell division also. So if you've seen, if you have gone through the schedule, I've kept this cell cycle and cell division twice, today and tomorrow, because of its weightage. Okay, it's quite important also because the cell division concepts would keep repeating in even 12th standard also. If you talk about meiosis 1, that prophase 1, meiosis 1, prophase 1, packaging, gygatin, leptotin, right? All these 5 different phases, subphases, all these things keep repeating. Okay, so you should be careful. Okay, so that's why I kept two. Just a minute, I'll be taking attendance. So meanwhile, what you can do is that's okay. 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 So I already mentioned, right? So whoever not taking the test and whoever not attending the classes, at the same time, whoever feels that mugging up is the best policy to study for the NEET, please quit the classes. You're not fit for NEET. I'm saying very openly, I'm saying because you can't crack the NEET if you keep mugging up. That strategy will not be working. Okay, whoever teacher says this, okay, mugging up will not be working. If you can really prove me wrong, I'm open to the challenge. Okay, so if you guys feel that mugging up is fine and you can go ahead with the mugging up whatever the chapters or the topics you read, these classes are not for you guys. At the same time, NEET is not for you. So keeping in that mind, if please don't waste my time and your time also if you believe in mugging up. Okay, right. So let's move on to the important topic. So you should be serious, guys. So it's almost 26th June means where you have 1 month. Imagine that there was no COVID. Okay, you would have been completed by now, and you would have been known your future by now, whether you got whether you would have been clearing the NEET or not. Okay, these words sounds pretty rude, but this is the reality. Okay, right. So let's move to the topic. So from tomorrow onwards, no more waiting. First 2 minutes, whoever comes, that will be attendance will be taken, and it will be posted in the group. Later will not be considered. That's it. Okay, let's move on. Coming to the unit of the life, that is cell, right? So here, how do we need? How we have to approach this chapter? Right, the cell, when we talk about the cell, we already talked about evolution already completed, and then I kept these lessons. Why? Because in an evolutionary perspective, you will be talking right from the universe origin, earth origin, and the primitive earth condition, and then you will be talking about the remaining aspects, right? So that's what I kept the evolution first. The plan which I gave in the schedule, NEET schedule, right? It is not just like randomly arranged. So please understand that. So you would have remembered how the first cell has origin, right? And then how the how this who is this the one who actually disproved this abiogenesis theory. This person doesn't have any evidence to talk about the how first cell has formed. So that's what we got. The Rudolf Virchow cell theory states that every cell actually comes from pre-existing cell, right? So these theories are important, and those bad evolutionary concepts are related to this. It means how the cell would look like, what are the characteristics of the cell, what are the cell organelles we see right within it, and what type of the cell organelles we will be having. It means there are single membrane bound cell organelles, double membrane bound cell organelles. There are cell organelles which doesn't have membranes also. These are the important aspects you need to remember. If you don't focus on this, you will be losing the marks. Okay, and if you that is one thing. Okay, so when we talk about the cell, we will be having different aspects that you need to cover. Is at least human cells you try to remember the cell shapes and cell structures. At least human cells, when human cells are concerned, immune cells, and what about neurons, right? Nervous system, and the units of the cells, units of this organ systems, right? So those things you need to focus. So we'll be talking in structural organization in animals and plants, where we talk about tissues. Now we are not talking about anything about tissues. So slightly focus on the human red blood cell. It is 5 to 10 micrometers in size. And what is the shape of it? Circular and biconcave. At least few cell shapes and their structure and their functions are important. Okay, when we talk about the cells, we already talked, we have different types of cells: prokaryotic cells and eukaryotic cells. What makes the eukaryotic cell eukaryotic and prokaryotic cells prokaryotes? So if you see the eukaryotic cells, right, um, you'll be having the membrane bound cell organelles. Like one of the important cell organelle that is absent in prokaryote is the nucleus, the nuclear membrane, right? So you have to focus on what are the differences between prokaryotes and eukaryotes. Okay, so in this chapter we'll try to deal with one by one. Means one cell organelle to other cell organelle we'll try to discuss. And regarding this cell chapter, I already took a few videos also. You can go through it. So when we talk about cell organelles, right? So first let's see about membrane bound cell organelles. So here we'll be focusing on what are the different types of cell organelles we'll see. For example, how can you guys say how eukaryotic cell origin? How eukaryotic cell origin? Yeah, Steve. Good morning. Or originated? How the eukaryotic cell originated? Any idea? From prokaryotic cell by evolution, sir. Yeah, that's correct, right? What is the theory behind it? Ma, whoever mugged up, na, they have to answer, right? You would have been having good memory of these things. Why you guys are not answering? Whoever believes in this mugging up facts. Yeah, what is the theory behind it, Steve? Have you guys remember the theory behind it is endosymbiotic theory? Endosymbiotic theory is a theory where it states that there is a primitive cell and there are some prokaryotes. So these prokaryotes, entered into this cell to become cell within the cell system, that is eukaryotic cell. So what is the theory behind it is endosymbiotic theory. Endosymbiotic theory. Okay, so that is the case. So with this, this is very essential because you need to know how the eukaryotic cell origin. Then you can really make these things will make sense. Okay, how come the cell is containing the mitochondria? You can't just like memorize things like okay, cell has mitochondria, cell has mitochondria, cell has chloroplast, cell has this, cell has that. But we have to ask one question that this is not the first time you guys are reading, right? So you have to ask the question. This 11th standard portion already happened. This cell structure you would have been answering right now. So you should be asking that how the mitochondria is present in this cell. If you can really answer, had the questions like that, then you will be able to remember for the lifetime. Okay, don't read for the sake of reading. Okay, so whoever likes it, you be in the class. That's it. So here we need to focus on one more thing, that is endosymbiotic theory, right? So cell within the cell system. So these mitochondria, chloroplasts, if you would have been remembering, what are the cell organelles which are semi-autonomous? semi-autonomous Semi-autonomous cell organelles, what are they? What are the semi-autonomous cell organelles? You guys have to tell. Semi-autonomous cell organelles. Name few semi-autonomous cell organelles. What does it mean actually? If you don't know this terminology, at the same time the concept behind this, you can't really go through. Very good, Kritika. Yes, Tasha Krishna, correct. Mitochondria. Semi-autonomous means basically within a cell, these cell organelles would be actually controlled by the nucleus, right? So they have no much individuality, right? And these cell organelles like mitochondria and chloroplast, they can behave by their own because they have their own DNA in it. Because once upon a time, back in time, during evolutionary process, due to endosymbiotic theory, these are the prokaryotic cell which is able to fix put this carbon or the sunlight. By using sunlight, which is able to fix the carbon dioxide. It's one of the prokaryotic cell, photosynthetic cell, which went inside the primitive cell and became eukaryotic cell, right? So likewise, chloroplast and mitochondria have been, or came inside our eukaryotic cells. No one will ask this type of questions. They wanted to, what they have to do is just memorize. So when you memorize, you can't really answer any type of correct or incorrect statements. You can't really handle that questions. And because if you does mistake in this type of incorrect or correct statements, you lose marks. Negative marking is there. Okay. So these are the important aspects you need to remember. One is endosymbiotic theory, you should know, because how the eukaryotic cell origin is what you need to focus. At the same time, what are the semi-autonomous cell organelles, right? And if you see this, more autonomy will be present in the nucleus, which controls the entire cell. So you need to know before you really read this chapter. Then you can start with the chapter with different cell organelles. For example, you will be having cell envelope. So if you talk about envelope, you will be having cell envelope and it will be modified. For example, how these cell envelopes will be present in prokaryotes and eukaryotes is really a big question. When we talk about the prokaryotes, right? Prokaryote has this cell envelopes, right? So if you remember this cell envelopes, right? They decide what type of bacteria they are, whether they are gram positive or gram negative as well. At the same time, they will be having chemically complex cell envelope, which consists tightly bound three layered structures. Okay, so if you see this, the structure of the prokaryotic cell, right? You'll be having three important layers. One, two, three. Three important layers, right? One is outermost glycocalyx, followed by cell wall and the plasma membrane. Plasma membrane is common for the eukaryotic and prokaryotic cell. So how you have to approach this lesson is, you have to make a tabular form and you have to write the important points, aspects, structural aspects of the prokaryotes and the eukaryotes. See which is common and see which are common and see which are not common. So then those questions can be of correct or incorrect statements. So these three layers within this prokaryotic cell, and what are the functions of this glycocalyx, what are the functions of this plasma membrane, what are the functions of this cell wall, you need to remember. So based upon the cell wall, right, we will be distinguishing this bacteria into gram positive or gram negative, right? At the same time, one of the important aspects or the important function of this glycocalyx is that it forms two types. One is thick layer or one is thin layer. So one becomes a slimy layer or one becomes capsule. So these are the two different aspects that you can see in the these glycocalyx, how the glycocalyx would be transformed. Either it can be loosely arranged or tight, thickly arranged, that is capsule form, and which really provides resistance against different drugs. So how far the prokaryotes are able to get resistance to different drugs? Okay, how far they are getting resistance to different drugs. So those resistance to drugs are provided by one of the layer of the layer of the prokaryotic cell. Okay, so that is one of the aspects you need to remember. So these are all the structural aspects that you need to focus on, right? And then what you can, and when we talk about the components of cell, when we talk about the cell wall contains few different layers actually, right? So again, in eukaryotic cell, again we have plant cell and animal cell, right? And prokaryotes as one type of, it is one type of cell. So cell wall of plant cells, how they are actually giving the shape, their functions also you need to focus, right? So here in this chapter we'll be more talking about the cell organelles and their associated cell organelles, at the same time their location and their function. Okay, so if you don't focus in that way, you will be losing. So how many times you revise, it will not be useful when you don't tabulate it and when you don't revise it properly. For example, cell wall of prokaryotes, cell wall of eukaryotes in the plants, how these cell walls would vary, what is the composition of these cell walls also you need to focus. Okay, and here in prokaryotes, one of the important specialized, right, special membranous structure, that is nothing but mesosome, right? What is a mesosome? How mesosome formed? From where, which layer of bacterial cell this mesosome is formed, and what is the analogous structure? It means that what it is, mesosome is equivalent to the one of the structure present in our eukaryotic cell. So the mesosome is present in the prokaryotic cell. It is equivalently having the same similar functions of one of the cell organelle present in eukaryotic cell. What is it? What is mesosome and how it is formed? Should be able to recollect, guys. So if you see this mesosome, they are actually infoldings of this plasma membrane, and they can form actually three different structures, right? So these extensions, if you see these extensions, right? So within this prokaryotic cell, right? So within this prokaryotic cell, if you take this is a prokaryotic cell, so within this prokaryotic cell, you will be finding these infoldings, right? So these are the mesosomes. So what exactly these mesosomes? So these mesosomes are special membranous structures, right, which extends to form vesicles, tubules, and lamellae. So what exactly their function is? Yeah, very good, Darshia. Correct. It's correct, Darshia. And they help in formation of cell wall, and even it helps in distribution of this when the cell is actually dividing, right? So even it would help in the distribution of this daughter cells, and even helps in DNA replication, right? Not only that, they also secretory in function, and even increase the surface area of the plasma membrane, because if they are infolding, whenever you see this infolding, even the brain has got this infolding, gyria and sulcus, so they actually increase the surface area, right? Like that, these mesosomes would actually increase the surface area of the plasma membrane, right? And when we talk about the bacterial cells, these bacterial cells can be motile and non-motile, right? They are of two different type of cells based upon the external appendages, like flagella. You will be having pili. The pili is a sex pili, or which involves in the sexual reproduction of this bacteria. So this bacteria can actually reproduce by sexually and asexually also. Sexually in the sense, sexually by transduction, conjugation, and transformation. Right? We have seen this transformation principle experiment, right? So in that we will be talking that two bacterial cells, for example, if you consider this is bacterial, this is bacteria. So these two bacterial cells, if you can see clearly, so these two bacterial cells, you see these are two bacterial cells, okay? So what happens is, imagine this is an axonal appendage. So this axonal appendage would be like this, okay? And so these axonal appendages would come and meet, and so that they can exchange the genetic material from one cell to other cell. So that's how this sexual reproduction takes place: transformation, transduction, and conjugation. Okay, I already told that I'm not explaining each and everything in this 1 hour class. Okay, I'll be releasing videos based upon your request. At the same time, what are the highlights that you need to focus is this pili or pili, and this flagella filament, you know, that if you remember the how these filaments are actually arranged, you would have remembered 9+2 arrangement, how this flagella, right? And even we'll be talking about this microtubule arrangements, centrioles. So those things, those aspects, arrangement, 9+1, 9+2 arrangement of this filaments that you need to focus, right? And then coming to the next aspect, that is when we talk about one of the functions. So what I can do is I will try to provide one PPT probably. Okay, in that I've already gave some uh, minutes. Yeah. So those are the one of the important aspects that you need to cover. Nothing but the components of the cell, the cell membrane. So these are external appendages actually. So apart from these three layers, you will be seeing in the prokaryotic cell. Not even prokaryotic cell, even you will be seeing this flagella in even sperm, which is a motile in nature. So let's move into the next cell organelles, like which are ribosomes and inclusion bodies, right? So these ribosomes, you need to focus on what are the types of ribosomes you see in prokaryotes and what are the types of ribosomes you see in eukaryotes, right? So these are the different aspects you need to focus on. At the same time, when we talk about inclusion bodies, you need to know what exactly they store. Okay, what are the reserve food materials, food reserves that will be stored in prokaryotic cell, and where exactly the location is at most important. These are all the things that you that are concerned when we talk about prokaryotic cell. So when we talk about the prokaryotic cell, you need to more focus on what is not present in eukaryotic cell. Like for example, you will not find this energy generating cell structures, because this prokaryotic cell itself is a unicellular organism. So within which you will be finding few of the cell organelles which generate energy. At the same time, what are the different layers that are present in the prokaryotic cell. At the same time, you will be focusing on each layer and their functions, and the exceptional case, the mesosomes, and the three types what it forms and what are those functions, right? And then shapes of this prokaryotes. At the same time, flagella, pili, these are the structures you need to focus in the prokaryotes. And even the ribosomes, what are the types of ribosomes that you will be seeing in the prokaryotes, right? So these are the aspects you need to cover in the prokaryotic cell. Is that clear? Are you able to follow what I'm explaining? Are you able to follow? Yeah. Okay. That's good. So next we'll move to the next aspect, right? That is, let's see each cell organelle how they have been arranged. Then we can get a clear-cut idea. Okay. I made one presentation actually. I'm looking for it that it can be used. I'll provide one link flowcharts. Okay, you can use that flowchart so that it will be more useful. Chapter 8, I already made few, what do you say, for your juniors, sub juniors. Okay, I'll provide that PPT in the class group so that you can access that. So when we talk about, what do you say, prokaryotic cell, so these are the aspects you need to focus. Let's move to the eukaryotic cell. What exactly eukaryotic cell? How do we look into eukaryotic cell? So in eukaryotic cell, we need to look three important aspects. One is plasma membrane or cell wall, nucleus, and cytoplasm or protoplasm. So what exactly the plasma membrane does? What exactly nucleus contains? Nucleus contains chromosomes. And then when we talk about the cytoplasm or protoplasm, again we'll be talking about cytoskeleton, cytosol, and cell organelles. So these are the three important aspects will be present in the cytoplasm or protoplasm. Cytoskeleton, cytosol, and cell organelles. So within this eukaryotic cell, within the cytoplasm, within the cell organelles, we'll be having three different types of cell organelles. One is non-membranous bound cell organelle, one is single membrane bound cell organelles, and one more is double membranous bound cell organelles. Right? So can you give examples of these three cell organelles, guys? Please give examples of these three cell organelles. One is non-membranous, next one is membranous, and the next one is double. Next one is double membranous. Can you give examples of each? No, NEET is something which we actually, when we're revising in the last neck of the moment, we need to recollect the stuff. And meanwhile, I'll be trying to provide the link for the, this this flow chart you can access and you can see, so that you'll be more clear. Okay. So please answer those questions, guys. Are you guys there? Please do answer. Meanwhile, uh, I will forward the PPT so that it will be more clear. What are ribosomes? Single membrane or double membrane? You can use code for non-membranous bound cell organelle NMBO, MBO, NMBO, DMBO. You can tell like that so that you can cut short the typing thing. Okay. Every time I'll try to make something easy for you guys and you will never understand. Anyways, so try to give in shortcut. That's okay. Membrane bonds, cell organelles, MBO you can give, NMBO, DMBO. So what are those? So what for this quick recollection, right? How much ever you memorize, that is a problem, right? So you have to have some memory technique. That's what I'm giving flow charts. Okay, I'm not completely against the memory technique. Memory techniques would work. I mean, memorizing would work, or mugging up would work only 1 to 10% only. So reminding, you need to have certain type of techniques. Now I'll be forwarding some PPT. Okay, then it will be more clear. Meanwhile, you please answer the questions. Yeah, right, correct, guys. Yeah, Mithula. What about others? So if you can't really have a sense of what are membranous and what are non-membranous and what are for example, what do you say? What are double membranous? How can you go ahead with the entire chapter? So that's what I stopped here. So I'm providing I'm providing one PPT so that if you go through that, you'll be understanding what I'm talking about. Okay. Fine. Yeah, that's correct. Correct. Now you just go through the link I'm gonna provide, so it will be. Actually, I thought you guys will quickly recollect. That's why I didn't arrange this flowchart first shot. But only one or two are able to recollect things. So trying to give the link for that. Okay, Arsha, correct. So what you guys do is in this link you will be having cell structure flowchart. Okay, you open that and you see, so that it will be very clear. What I'm how to approach this lesson would be very clear. Okay, so let's move on to the next aspect. So each cell organelle will try to look into. So within non-membranous, when we talk about single membrane bound cell organelles, they are Golgi bodies, endoplasmic reticulum, lysosomes, vesicles, and peroxisomes, right? And within the endoplasmic reticulum, again we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Clear? And then when we talk about non-membranous bound cell organelles, we have ribosomes, microtubules, and centrioles or centriosomes. So this entire flow chart, I made a beautiful flow charts you can go access. And the later part is double membranous bound cell organelles like nucleus, mitochondria, and chloroplast. Is that okay? And when we talk about one of the important aspects you need to cover is endomembranous system. What is endomembranous system? Here we talk about endomembrane system is we'll be talking about so endomembrane system consists of few cell organelles, right, which actually work together. They are, if you see, they are endoplasmic reticulum, lysosomes, and vacuoles, and finally Golgi bodies. So within this endomembrane system, the few proteins are which comes right from the few proteins would be processing. At the same time, you can see that they will be involved in transport of some biomolecules. So right from the endoplasmic reticulum and then lysosomes and vacuoles and Golgi bodies, all these four together, we call it as endomembrane system. So in NEET you may get questions like they may alter the options of the cell organelles, and they may pick the endomembrane system cell organelles. So you should not be getting any confusion while you picking the correct answer, right? And then let's talk about the cell membrane. So cell membrane is one of the important membrane which actually has certain integral proteins within the cell membrane itself. Just like our room has some doors and windows, it will have integral protein. At the same time, it will have sugars also, and it is made up of lipid bilayer. And we have a fluid mosaic model. Why we call fluid mosaic model? At the same time, who gave the fluid mosaic model? You need to focus. And here we'll be talking about the passive transport, where there won't be any involvement of energy to actually diffuse certain molecules right from the external environment to the cell, or cell to the external environment. So simple diffusion may occur, and selectively they will allow certain molecules through this plasma membrane. So these are the things you need to focus. But at the same time, in the plasma membrane, if you really remember the structure of the the axon we talked in the neural coordination, right? So if you remember the neural coordination lesson, you'll be finding axon, right? Axolemma contains certain proteins, right? So which allows N plus and K plus ions. This sodium and potassium pumps, which actually pump this sodium and potassium actively. It means it involves the ATP utilization. So active transport, passive transport, these all diffusion, osmosis, we'll be talking more clearly. I will be explaining on the board in the next class. But you have to remember that passive transport doesn't require any energy, but active transport requires energy, and that will be given by ATP, and these ions would be transferred from outside environment to the inside by this pumps, proton pumps, or by this ion pumps like sodium and potassium pumps, right? And then let's talk about the cell wall. Cell wall, when we talk about the cell wall, we have different type of cell wall we'll be seeing in the what do you say, the first cell wall was discovered by actually Robert Hooke, right? So he talked about the cell wall. So the cell wall composition and where exactly present. So if you see this typical cell wall may hold four different layers. They are middle lamellae, primary cell wall, secondary cell wall, and tertiary cell wall. So each of this cell wall and their components and their composition and their functions are important. For example, if you see primary cell wall, they are thin and elastic, right? So you need to focus on what type of cell wall we are talking about. Within the cell wall, there are other few structures out there. What are their functions, right? So here plasma membrane is this lesson you need to approach as their functions as well. What are the functions of the plasma membrane that we need to talk about? And when we talk about the double membrane structures, we will be talking about the nucleus. So how do we approach this nucleus? So this nucleus contains two important components. One is nuclear envelope or membrane, or nucleoplasm. So within this nuclear envelope or membrane, you will be finding outer, inner nuclear pores. You have outer membrane, inner membrane, and nuclear pores. And the outer membrane, actually the foldings would form the rough endoplasmic reticulum. So it would be joined, and finally it forms rough endoplasmic reticulum. You know why it is called as rough endoplasmic reticulum? Because it contains what it contains. It's a 6th standard question, right? So Ribosomes it contains, that's why it is rough endoplasmic reticulum, right? And then we have nucleoplasm. In the nucleoplasm, we have nucleolus and chromatin. So these are the two important components you need to focus. So within the chromatin or chromosomes, right, this chromatin or chromosomes are made up of three important components. One is DNA, genetic material, and associated histone proteins and non-histone proteins. I gave very clearly in the flowcharts. You have to go through that, right? So here packaging we already talked in molecular basis of inheritance. So we don't need to talk much about packaging here, but you need to focus on what do you say, these what do you say, what are the functions of this nucleus. So nuclear pore, what does it do, and how nuclear pore formed? It's a, it is an integration of both outer and inner membranes, and finally it would be pinched over there and it will form a pore like structure, right? And then when we talk about the mitochondria, we have to focus on its structures, inner and outer membranes, and what are cristae, right? This cristae contains this ATP generating proton pumps, ATP synthase, that will be talked in the plant physiology as well. So what matrix contains, what cristae contains, what is inner membrane space, it is present between outer membrane and inner membrane, right? At the same time, this cellular respiration is under the control of this mitochondria, right? And what are the raw materials? For example, it takes up the glucose and it actually oxidizes it, and then it releases the energy in the form of ATP. So you need to focus on this F0 and F1 particles, where exactly they present, in which parts, cristae are, or they are present in matrix are, inner membrane, outer membrane. These are the questions you may utterly get confused. So you should be practicing by diagram. Okay, diagrammatic practice is needed, right? And then when we talk about the plasmids, we have different types of plasmids, right? So when we talk about the plasmids, we have chloroplast, leucoplasts. At the same time, we have chloroplasts. So each plastid has got its own function. For example, chromoplast gives the color. For example, yellow or red in color. If you see the cells or some plant tissues, you will be finding that they are in yellow color or or orange color, because presence of some pigments, right? Those are carotenoids. Next, leucoplasts, which are the plastids, are actually colorless and their function is different. So what you have to do is make a flowchart while you're reading, right? Plasmids. What are the different types of plasmids? You have chromoplasts, leucoplasts, and chloroplasts. Within the chloroplasts, again, what are the different types of pigments and their wavelengths also you need to focus. Absorption wavelengths. And if you take the chromoplasts, they have they have different types of functions when compared to leucoplasts. So they will be having absorption. For example, they found in fruits, flowers, and leaves. So they give the color. And leucoplasts are absolutely colorless, and they generally occur near the nucleus, right? And they store food materials. And everyone knows what are the functions of the chloroplasts. And if you see this chloroplasts, they are double membranous structures, and they contain thylakoids and within the stroma. So, and then when we talk about the endoplasmic reticulum, we have two different types of endoplasmic reticulum: rough endoplasmic, smooth endoplasmic reticulum. Why it is called rough endoplasmic reticulum, and what is its function in this endomembrane system? So when we talk about endomembrane system, right? So when we talk about this vesicles, Golgi body, this what is endoplasmic reticulum, what exactly their role, how they form vesicles, why these vesicles are very important. So if you see these vesicles, almost in 2013 or 14, we got Nobel Prize in medicine when we talk about these vesicles, how this transport of vesicles are taking place right from the nucleus to the cell membrane. So that is one of the intruding or one of the important aspects you need to cover. So when we talk about the endoplasmic reticulum, its structure, where it is present, its functions, just like that. Like that, you need to approach each of the cell organelle. Are you able to follow, guys? So this is very simple lesson. According to NCERT, if you would have seen in NEET book, it would be a ocean actually. It's an ocean actually. Each cell organelle I can speak for a day, right? But here, in exam perspective, you can stick to the NCERT, right? Line by line you read at the same time. If they are focusing on certain highlights like chloroplasts, means you need to go back to the other lesson which it contains about, which talks about its pigments also. Don't just ignore just like that. Okay, so when we talk about this leucoplasts, how they are actually storing the food materials, on what, in what form they are storing the food materials, also important. So the potential questions might be like amyloplasts, eleoplasts, and eleroplasts, right? So for example, my question is, in leukocytes, this leucoplasts, in the leucoplasts, amyloplasts are there. What amyloplast would store? What kind of a substance? What are the organic molecules it will store? In what form it will store the food material? Try to tell. Amyloplasts. So this is a potential question within the plastids. You'll be finding different plastids, right? Amyloplast, eleoplast, and you have what is a eleroplasts, right? And their storage molecules. So this can be a column A, column B type of questions. Arshia is saying amyloplasts, actually stores carbohydrates in the form of in the what form, Arshia? Actually, yeah, in the name itself is a amylase activity, right? We have talked in 12th standard practicals also. So they cleave this carbohydrate, especially in the form of starch. So likewise, you need to focus on these plastids and their storage food materials. I hope you are getting what I'm talking. Like that, ribosomes. What are the different types of ribosomes? So if you take 80S type and 70S type, where they present in prokaryote cell, eukaryote cell. So those things you need to focus. When we talk about the cytoplasm, cytoskeleton. So within this cytoskeleton, the arrangement, right? The diagram that was given in the NCERT, you need to focus on. That is, internal structure of the cilia or flagella, and how this axoneme is arranged, or what is the type of arrangement you see in this cilia or flagella. So you can see that 9+2 arrangement will be seen in this flagella or cilia, and how this is different from the centriole, that you need to focus. So when you see the centrioles, it has slightest deviation right from the cilia or flagella, right? So for your juniors, I took this chapter, so they are much clear about that. We had a separate diagrams for both of them. It means when we talk about the cilia and flagella, and when we discussed about the centrioles, there is a difference between this fibril arrangement, right? So that difference is important here. So in centriole, you will find triplets, right? Triplet arrangement will be there. But when we see in this cilia or flagella, you will be finding duplets arrangement. So douplets, the microtubules would be arranged as douplets or duplets. Yeah, very good. So these microtubules will be arranged as 2 in number in cilia or flagella. Right? So this is a potential difference you need to manage. So, and their functions, where exactly this 9+2 arrangement is there, what are the functions of these centrioles, and how microtubules, how they are very essential during the cell division, that you need to focus, right? So when we talk about the when we talk about the chromosome in the PPT, I already gave different types of chromosomes. Shapes of chromosome would form during the cell division. Those are metacentric, submetacentric, acrocentric, and telocentric. So these are the different types of cells. So they may be giving column A, column B. They will be giving that the centriome location middle, between the middle or end, or close to end, or at the end. So like this, they will be giving the position of the centromere, and they will be giving the names of it: metacentric, submetacentric, acrocentric, telocentric. So you should be able to match it properly. Okay. And here when we talk about this chromosome structure, right? So in the chromosome structure, you need to more focus on the centromere. So centromere and its location is more important. And what are satellites, and what are their functions, and what are microbodies? For example, microbodies are minute vesicles, right? And they actually contain certain enzymes both in plants and animals. So if you go through this NEET material or NCERT, you will find these are the cell organelles which are covered in NCERT. What you can do is take the cell organelles first, and next column you can do like this actually. So what you can do is you make a tabular form, but in NEET book they already gave very clearly. But if you don't have NEET book, that's absolutely fine, no problem. And you have NCERT, that is enough. So what you can do is name of the cell organelle. For example, nucleus. You take nucleus, and then you talk about whether it is single membrane or double membrane or non-membranous, and then location. Talk about location, right? And its structural components. So what are its structures and their functions? So like that, you can make a table of each and every cell organelle. So in one place you will get a big table. That would be clear enough. So that is it for today's class, where have you guys accessed that file, link? The link that I provided, did you guys access? Did you guys access? There you can find other PPTs for other lessons also. You can actually access. Did you guys access this link? Are you guys seeing? Yeah, very good. So as this lesson is very small, but the approach I already gave in the PPT. So now you can start after the physics class. You can go through your lesson. See that lesson in what way I have shown you. In the sense, the flowcharts way is the best approach you can have it, so that you will have you will not be having any sort of confusion. How much ever you try to revise the same chapter for 1,000 times, if you don't have a proper strategy, you will not remember. You will not recollect. More than remembers, you need to recollect. So please do follow that flowcharts, and please work if you have doubts, you can ask me. Okay. At the same time, please read in that format. So we'll meet you in the next class. In tomorrow's session, we'll try to deal with that cell division and cell cycle. Okay. Once you finish this chapter, straight away go to that chapter. Don't waste time. Thank you, guys. Yeah. See you in the next class.